group of researchers, scientists studying plant biology, how our brain forms and keeps memories, how the human body functions and how it combats diseases like cancer. This is one of their stories.
Hi, my name is Maria. I was a high school scholar at the Salk Institute. When I first arrived at the lab, I was shocked to see how many people there were, each of them working on their own unique projects that seemed to fit together like a puzzle. My lab's main focus was to understand how DNA influences disease, and it was led by Dr. Inder Verma. I learned that the very genetic code we are made of can also make us sick and even cause cancer. During my time at the Salk Institute, I worked with Dr. Amy Rommel, and she focuses on the most deadliest form of brain cancer, glioblastoma.
What was it like working in a lab for the first time?
All I know is that I am forever changed. My eyes are now open to all the excitement that goes into research.
Hi, my name is Ben, and I was also a high school intern with Amy at the Salk Institute. During my time in the lab, I learned that a lot of planning goes into each step of the scientific process, and that everything has a clear purpose.
Okay, go right ahead.
Is this thing on?
Yeah.
Okay, good.
Tell us a little bit about your background.
So I'm from Dallas, Texas, a town where our high school mascot was the Fighting Farmers. So you can imagine I grew up in the country. I grew up always loving science, but I didn't know I could actually make a career doing it. Then I went to college and met some researchers doing incredible work. I volunteered for a few years before being taken on as an employee in a research laboratory, all the while I was still finishing my degree.
So, what brought you to the Salk Institute, and why did you go into brain cancer?
So that all started when I was in Dallas, Texas. I was a graduate student working to try to figure out how to help patients whose breast cancer became resistant to the very drugs we were trying to treat them with. I read several papers on how tumor cells can adapt by changing their shape, the way they move, and their genes. I even attended specialized conferences where scientists would present their most cutting-edge research. But I was struck by one particular paper coming out of the Salk Institute from Inder Verma's laboratory. He's world-renowned in the field of gene therapy. His laboratory actually discovered that when we treat patients with a drug that can cut off the blood supply that feeds the tumor can come back, and it comes back resistant to the drug. In fact, it creates its own blood vessel cells. I immediately thought, if I can purify these cells and find their weakness, maybe I could develop a way to kill them. If we could do this, then the tumor would not have access to the normal blood vessels, and it wouldn't be able to create its own. And obviously that's how tumors can grow. They need nutrients from the blood. But before doing this, I had to discuss it with the lab. Funny enough, the best place to discuss new scientific ideas is at the lunch table. I decided the first step I needed to do was purify these tumor-derived blood vessel cells.
What is the experimental question?
My experimental question was, what is the tumor blood vessel cell's weakness? So I could try to figure out how to kill them and starve the cancer so the cancer can't survive. To do this, the first experiment I needed to do is:
What is a logical first experiment? A. See if tumors can create their own blood vessel cells. B. Purify the tumor blood vessel cells. C. Look at the DNA of the tumor. Or D. See how long I can balance a spoon on my nose.
The correct answer is B. Purify the tumor blood vessel cells.
Amy told me that the first experiment she did was to purify tumor blood vessel cells. She needed to purify them from the rest of the tumor so that she could analyze their RNA.
What is RNA, and how is it different from DNA?
Let me get Amy and Ben. They'll help me with this one.
The best way to explain the relation between DNA and RNA and protein is to look into one of my favorite systems, paper airplanes. The internet has all these blueprints for a paper airplane. It stores all of this information so we can access it when we need it. That's exactly how DNA works. DNA is stored deep in our cells and contains all the information that makes us, well, us. If I find, for example, a paper airplane design that I really like, I can make a copy. If I do this to a gene, it's called transcription in the cell. Importantly, this copy is not yet an airplane. It is just a printed blueprint that I can use to make an airplane. This is exactly how RNA works in the cell. RNA is a copy of a gene, but it's not actually useful yet. The real workers of the cells are the proteins. These proteins are made from the RNA. In the cell, the RNA is turned into amino acids, the building blocks of protein, and then folded into functional proteins. This is called translation.
Now our paper plane flies. So the DNA is the long-term storage of the information in our body. The RNA is a copy of what the cell wants to make.
And the protein is what actually does the work in the cell.
Exactly. We are going to purify the tumor blood vessel cells, analyze their RNA, since that is what ends up becoming a protein. That way we can find their weakness.
And kill them.
What is the process called where DNA is turned into RNA? A. Biology, B. Translation, C. Conformation, or D. Transcription?
The correct answer is D, transcription.
Inside this box, I have a sample from a patient's tumor. So what we're going to do is we're going to use a specialized protein called an enzyme, and we're going to dissociate the tumor, which is right now a clump, into single cells. Then, once they're single cells, what do we want to do? We want to pull out our tumor-derived blood vessel cells. So in order to do that, we want to basically, like fishing for a fish, we want to isolate the cell surface proteins that are just on the cells of our choice. So we just pull out the tumor-derived blood vessel cells. So that's what we're going to do.
What do we absolutely need to purify a specific type of cell? A. It needs to look different to the eye. B. It needs to have unique markers inside it. C. It needs to have unique markers on the outside cell surface. The correct answer is C. It needs to have unique markers on the outside cell surface.
Once we have finished labeling our tube of tumor cells, they now express different colors of fluorescent proteins depending on the type of cell that they are. This machine is a specialist in purifying tumor-derived blood vessel cells. We want to purify the green fluorescing population of cells, and these lasers can actually kick out these different types of cells into their own individual collection tubes. All right, so inside this tube, we have purified our tumor-derived blood vessel cells. They're really, really small, so we can't see them. But we need to isolate the RNA from these cells so we can find their weakness. So that's what we're gonna do now.
What did Amy forget to do for this experiment?
Amy forgot to include controls.
Oh wait, we forgot something, you guys. See, not only did we purify the tumor-derived blood vessel cells, but We also purified the normal blood vessel cells. These are called our controls. We need something to compare our data to, so that's why we have to purify both, so we can have controls.
Now that Amy has her purified cells and controls, what will she do next? A. Analyze the DNA. B. Analyze the RNA. C. Go get lunch.
The correct answer is B. Analyze the RNA.
Wait a little while to get my results back from RNA sequencing. So, time for lunch.
Let's recap. What are some good controls for Amy to use in this experiment? A. Tumor cells. B. Tumor-derived blood vessel cells from more than one tumor. C. Normal blood vessel cells. Or D. Normal brain cells.
All of these are great controls, but Amy is only using the first three in her experiment.
I first take all of the data and compare my experimental results, the RNA levels from the tumor-derived blood vessel cells, to all of my controls. I then make graphs to represent the data and look for trends. For example, in one comparison, I noticed that the tumor cells express a certain pathway involved in heart formation very, very low. But when these tumor cells become blood vessel cells, this pathway is increased. It is possible that the tumor needs to increase the levels of this pathway in order to push the tumor cells to become blood vessel cells. But I won't know this for sure until I test it in the lab. I can't just start testing all of these genes because there are hundreds of them that seem to be altered in my process. So I need to read a lot of other scientists' work to take my very long list of genes and slowly decrease it down to a manageable list that I can test in the lab.
How much do you think it costs to sequence an RNA sample? A. The cost of a cheeseburger. B. The cost of a toaster oven. C. The cost of a family of 3 to go to Disneyland. Or D. The cost of a car. The correct answer is C. The average cost to sequence one sample of RNA is about $500.
So now, because of our reading in the literature, we've narrowed down a list of hundreds of genes now to just 5 we're going to test in the lab. So now we need to do a very inexpensive experiment to see if any of these genes are important for tube formation. One of the cheapest experiments is called the tube assay. And what happens here is when a tumor cell, or when any cell, acts like an endothelial cell or a blood vessel cell, it can attach to a gel matrix and form these projections. And those projections can form these tube structures between the other cells. This is how you know that the cells are becoming blood vessel cells.
And they are glowing green because those are the tumor markers. So we know they came from the original tumor.
Maria, that's exactly right. These tumor cells were labeled with GFP a long time ago. That's why they're glowing green. And even though they become a blood vessel cell, you can tell they've still come from the tumor cells originally because they're maintaining that green glowing color. if we inhibit one of these genes on our list and it has nothing to do with this process, we should still get these green tube-like structures. But if the gene has an important role in converting the tumor cells to the tumor blood vessel cells, then we would block it. In other words, the tumor cells would not be able to form these green tubes like this. We perform this experiment by knocking out these genes one at a time to find out which one may be responsible for the ability of the tumor to become a tumor blood vessel cell. These are our results.
If this is Amy's data, which gene or genes could be responsible for the ability of the tumor to convert to tumor blood vessels?
The correct answer is gene F. Loss of gene F was enough to prevent the tumor from being able to create its own tumor blood vessel cells. And in case you're curious what gene F is, it is actually part of a family of proteins called semaphorins. These semaphorin proteins are involved in patterning blood vessels, and it's known to do this in embryogenesis and in heart formation. So it makes sense that it would also be involved in patterning blood vessels for tumors. We have successfully purified the tumor blood vessels, performed a genetic screen, and taken a list of hundreds of genes down to 2 that seem to be important in this process.
What is the conclusion of this experiment? A. Tumors can make tumor blood vessel cells. B. Gene F is important in creating tumor blood vessel cells. C. Gene B is important in cancer. Or D. Science is awesome.
The correct answer is B, but D is also correct.
Science is awesome.
Yeah, that must be the answer.
Well, you're both correct. And we were actually able to find out that gene F is important in the ability of the tumor to create its own tumor blood vessel cells. This information helps me to explain how the tumor adapts when it is starving by creating its own tumor blood vessels. My next step with this project will be to see if I can block the ability of the tumor to create these blood vessels by blocking this pathway that we just tested, and hopefully it will starve the tumor. I hope this result is effective in killing the tumor. Now it is time to talk to other scientists about these exciting results and get their advice and maybe forge a new collaboration.